Yes. Let me just go over one more example. Okay, so here we go. We're gonna do this example here. This example? Yes. Okay. Just scoot yourself, sorry. All right, so here we go. We're doing the same thing we just did. Follow these steps that we have on the board. Okay, this one you're gonna have to do all the steps, I think. Yes. So our first step, we need to find the field of view at low power. Wait. Actually, I like this other. We haven't done this one yet, right? Okay. Yeah, we have. No, it's not the pig nose one in your notes. It's slightly different. Because this looks exactly like what's going to be on your test. Okay. So we have to start with our first step. So how do we find the field of view at low power? First, we have to read what? The millimeters on the ruler? Yes. Okay. So what would we say the field of view about would be? We're going to go with 90 millimeters. Okay. So we do 90 millimeters. Well, it's just 90 millimeters at low power, right? So number one, our field of view at low power is just 90 millimeters. That part's easy, right? Number two, now we need to find our field of view at high power. So now we need to use our equation. So we're going to do, all right, I heard one person. Do we agree with that? We do our low power stuff over our high power. So our low power is 90 millimeters times what low mag? 15 divided by our high power, 50. Somebody do that, please. Oh, no. I don't know if the recording will catch this, but maybe. What? Even? 27 what? Millimeters. Okay, so we've done step two. Now we know our field of view at high power, which is the first answer. Okay, our third step, what do we have to do? How many objects would fit? Yes.
3.
Oh.
2.5.
It tells you the low mag right here, 15. 3. And then the high mag is 50.
2.5. No,
okay, so remember, we're not doing the red circle. We're doing the actual clump of cells. Okay. If I need you to do the equation, I have to give you the low mag and a ruler, or you will have a ruler on your test. And then I have to give you the high. Okay. So we're saying how many would fit? I would say it's hard because I don't want to draw on it. I hear your 2. I think I might up it to maybe like 2.1 or 2.2. That is the plan. Yeah. Don't worry. Don't worry. We have a plan. Your test is mostly going to be this. Okay? That's why we're going over it a lot. Okay. So I'm going to say, let's just go with 2.2. On your test, it'll be easier because you can actually draw the circle, right? And on your test, I'll have a range. Okay, so if you went with 2 or if you went with 2.2, either way, I'll give, you did it correct. As long as you're doing the correct answer. If you put that 7 would fit there, obviously that would be wrong. If you're close and your answer makes sense, it'll be fine. Okay, so I'm going to say that 2.2, we're going to call that a clump of cells, 2.2 clumps would fit in the field of view. So what's my equation that I use?
27.
27, our field of view, 27 millimeters times what fraction? 1 over 2.2, because I know that 2.2 would fit, but I only want to measure one of those 2.2, right? So 1 over 2.2. So what is that? 27 divided by 2.2. Sorry, one person. 12.27. If you forget your units on a test, you will lose points. Millimeters. Very good. So we have our sample length in millimeters. So we had our first answer, our second answer. And the last thing we have to do is convert millimeters to micrometers.
12,000.
So, yep. So we move our decimal 3 times to the right. 1, 2, 3, add a 0. So it'll be 1227 micrometers. Somebody in biotech called me out for not putting my commas. Does that bother you guys?
No.
Okay, it's the same thing, right?
The commas actually bother me more.
Okay, I'm sorry.
No.
All right. You ought to test.
I forgot the 0.
It's
Oh.
1200.
That's ugly. I flunked the test right there. Hopefully you guys will do better. On a test, you can put the comma or not put the comma, as long as you put your comma in the right place. Okay. Are we feeling a bit better on understanding this process? Okay. What we're gonna do now, we can do another one at the beginning of tomorrow. You're gonna get, half of you will get on microscopes, or 8 of you will get on microscopes. And you're gonna either finish your half sheet, and because you are turning that in, okay, you need to have your name on it, right? It's over here. Name, the field of view at low power, which we know is already done. Fill that in. Everyone's got 4.1. Field of view at high power. Your diameter in millimeters, your diameter in micrometers, what you were measuring. So draw your sample. Tell me what you were measuring. What is your sample? Is it frog's blood? Is it a mosquito? And then I might even see on your drawing, I might even see circles. That's not the same thing. Sorry. If I see circles, that's great. That shows me that you were doing the work. Okay. Good. I would do midnight.
I tried. It's still hundreds. Oh, yeah.
You can estimate. This whole standard is estimating the size of an organism. So if you can't draw that many circles, you can use your like mental math to say, I think about 1,000 would fit. Okay, that's fine. And then your fraction will be 1 over 1,000, if that's what you think. As long as when I look at it, it makes sense. And I'm like, yeah, I can see 1,000, and we'll be fine. Okay, is anybody completely done with their hash sheet? Okay, so we are gonna have to move by task. So if you're on a microscope, you're doing microscope. If you're not, we're doing notes. Okay, so let's get 8 microscopes out. Let's start with the people who are sitting at those desks. Yes. I have to look at the microscope.
100.
Oh yeah, you guys can play with taking pictures of what's there.
Oh, yeah.
Diameters?
Hard to read. Blue one. What's the blue one?
That'd be low
40?
powered.
Yeah, so the highest is 100.
What's the lowest magnification?
That's not the FOV.
No. No, the FOV is 4.1. Yes. The lowest magnification. 4.1 times. 2 days on Tuesday, let's
You need to use that to find your field of view at low power. Does it look like 4. Okay. So that's your field of view at low power. 4. And you have your. Okay. Then what magnification did you do this on? The next one. So you did that at 10. Okay. So now you need to use the equation. Your low power field of view times your low power mag over your high power mag, which was 10. Yep, 10. Right, so you did 4 times 4?
Yeah.
No, because your low power mag is 4. But this may not be true for you guys because you guys may not have done a magnification of 10. If you did a magnification of 10, this would be true. Yes. Okay, let me get you guys to take some notes and then we're gonna come around, we'll do something. The slides are in the cabinet, the wooden box and the grayish box. And then your plugs, you guys got those out of the bag? Okay. Let's make sure all microscopes are being used. We have 3 more microscopes. Somebody get up and use them. We can't do this all day. Huh? Highest mag is 100. Yes. If you want to use the picture, that's fine. Do you know what magnification you use? You went up 1? So 10. Okay.
What's the highest?
The highest is 100. Yes.
Oh, on the thing it said paper 1. Doesn't need calculators, but paper 2 and 3 is fine.
Okay, yes, you will have a calculator. Thank you. I've been meaning to look that up.
Or you can just do a multiplication.
You could. Okay, you guys, if you are not on a microscope, we're going to take some notes. So you should be on this page of your notes, please. The next page of your notes if you are not on a microscope. Wonderful. Now I have a recording of you singing. And it'll be posted on YouTube. No. No, not my mammal spinal cord. Okay. Are you on this page? Guys, by the way, still have 3 microscopes. They're being wasted. If you are not done with your drawing, you need to be on a microscope right now. And one of these 2 desks. Sorry, we only have 2 tables, but grab a microscope, get to a spot. Yeah. Yeah. As long as we have word definition of it.
No, that's like
And,
high power.
yeah.
Oh, and then, okay, no, then you do.
What is that?
What was the 3rd highest mag?
So you need a draw. The 3rd highest?
Yes.
So it goes 4, 10, 40. All right, are you guys on this page? This page. Okay, you guys need to be ready to write on this page. Ready? This page? Okay. Oh, nice. Wonderful. Okay. If you're on a microscope, either talk very quietly or not at all. Keep an ear open, okay? But focus on getting that half sheet done. So drawing is a very important skill in biology and is considered a type of data collection because drawings help to record data from specimens. So a drawing is the result of a long period of observation at different depths of focus at different magnifications. So what you did on your half sheet was not an actually like full IB biology drawing. What we did on the half sheet, I would call a sketch. Okay, a drawing has more rules to be successful. So you did a quick little sketch. Now we're going to talk about how we do like a formal drawing. Okay, so in your data table here, you have these things that you need to write down. Okay. I bolded the main parts. You do not need to write all the sentences. You might read the sentence, and if something sounds important, add that, right? But the bold is the bare minimum that you need. So for drawing materials, you have to use a pencil on white unlined paper. So if you are asked to draw something on an IB exam, they will have a blank spot of paper, and that's where you will draw. And if your pencil is not sharp, you will raise your hand and say, I need a sharp pencil. Okay? Just like any of your other SATs and stuff, all have pencils. So you cannot use a pen. All right. The positioning has to be in the center of the page. Okay? So you want to shoot for the center, not only because that's a rule, but also because we are using labels. You have to put it in the center, leaving room for labels. The size, it should be about a half a page. So when we are going to do a drawing, an official drawing that's different than the sketch you just did, I will give you a full sheet of paper, and you will draw in the middle, and you're going to shoot for about the size of a half a page. Okay? So you're not going to draw it on the top half. You're going to draw it in the middle. Ian, are you done with the microscope? Okay, then let somebody else do it. Who needs to use the microscope? All right, Ariana, go ahead. All right. Labels. You will use a ruler. I should have ruler bolded. I will do that right now. Labels. You will use a ruler to draw straight horizontal lines. They have to be horizontal. Okay, no angled lines. Don't worry, I'll show you examples. Yes. All right. Labels should be printed, not cursive. Sometimes when we do nice drawings and things, we want to make it look fancy, but the rule is no cursive. Oh yeah, put that on. That's no longer your picture. Okay, so we're using a ruler. Yes. I would say for this, do your best to write straight, but as long as you're not doing like cursive letters. I feel like yours is printed, but it almost looks cursive-y because you kind of have curlies in there, but it's not actually cursive. But as long as it's printed, I think you'll be fine. Technique. Lines are clear and not smudged. So you know how sometimes you're drawing in pencil and your hand goes over it and it smudges it? You want to not do that. Okay, clear, not smudged. Avoid feathery pencil lines and gaps. This is why we're going to use a sharp pencil. Feathery, like instead of, if I was going to draw like a circle, I'm going to make sure it's like a solid, good circle. Instead of like, it's kind of already feathery on here, but like, you know how it's like not, I don't know. It's hard to explain. But I'll show you examples. Yeah, don't do that. There are almost no erasers, I don't know, or stray marks on the paper. Color is used carefully to enhance the drawing. I think on the IB exam, you don't need to use color, okay? Stippling is used instead of shading. I'll show you an example of what that is. If you are in art, you might already know what stippling is, but it's basically dotting versus shading. Okay, so I'll show you an example. All right. Are we good on this?
Yeah.
Accuracy. You need to only draw what is seen. If you're drawing something and you know what it looks like, but you're not seeing it, don't draw. It's only what is seen. Avoid making idealized drawings. Do not necessarily draw everything that is seen in the field of view. Okay. So if I show you a picture of a bunch of cells and I say draw the plant cell only, you're going to draw only the plant cell. Okay. Or like that last example that we did, like with the clump of cells, I would say draw that clump of cells. You wouldn't have to draw all the cells in that, right? You're just kind of with your sketches too. You might have just drawn a piece of what you were seeing, not the whole thing. Draw only what is asked for. Show only as much as necessary for an understanding of the structure. A small section shown in detail will suffice. If it is time-consuming and necessary, for example, to reproduce accurately the entire contents of a microscopic field. When drawing low-power plans, do not draw individual cells. Okay, if you're drawing a mosquito on low power, you're just going to draw the shape of the mosquito, maybe the eyes, maybe the antennas, maybe the legs. You're not going to draw every single cell of the mosquito, right? Okay. Show only the distribution of tissues. When making high-powered drawings, draw only a few representative cells. Indicate thickness of walls, membranes, okay? Basically, just draw it exactly like you see it. If you do that, you'll be fine. Title, we will always have a title. Just like a graph, your title is very specific. So your title should state what has been drawn and what lens power it was drawn under. I should actually have that on this half sheet that you're supposed to be giving me. If you guys could add that, that would be wonderful. Your sample name, what did you draw? And what was your magnification? Was it 100x? Was it 40x? Was it 10x? Okay. On an official IB drawing, you will have to tell me that. So you might say, frog blood cells at 100x, for example. Somewhere in there we have a frog blood smear. So, for example, phrased as drawn as seen through 400x magnification. Again, I'll show you examples. Title is informative, centered, and larger than other texts. Title should always include the scientific name, which is italicized or underlined. So, if you are terrible at writing italics, I don't know if you've ever tried it, like trying to write italics. Some of us are terrible at it. So if you're terrible, you can underline it. Or if you already write kind of italic-y and you don't think you can go more italic-y, then do the underline. Okay. Remember, as soon as you're done with your half sheet, we need to give the microscopes up to those who need it. So if you are done with your half sheet, you need to leave the microscope. Yes. Okay, so field of view on high power. It should be smaller, right? So you did 4, so you did 6 minutes. We need to figure out. We need a whole thing. Okay. How many of those? One.
I don't love everybody. Everybody. I don't have to create.
Okay. But you have, so 1.3
You know
would fit,
exactly
right?
how to get rid of them.
So that's our fraction. So we're going to do our field of view at high power, which you said was 1.6. And we multiply that by our fraction. So you only want one of those 1.63. So one of the 1.6. You're gonna do 1.6 by 1. Well, times 1 over. Yes. I would go up to to the right. Yeah, that would make sense. Of course. Yes. Okay, so 4, we need it on 10 mag. Okay. Diameter. Wait. Your diameter and your millimeter should not be the same as your field of view. Did your cell take up exactly your field of view?
No, I didn't.
You have a 1/7 here. Does your organism take up about 1/7 the field of view?
I know. Part of it's all come together. That's a bunch of.
Okay. So ideally, you're gonna want to draw the circle, right? Draw your field of view and then draw your thing inside of it, so I can see the perspective. So you're just doing that kind of like butterfly thing. Yeah, so you have that drawn, right? You need to draw your circle around it so that we can see how many would fit in there. Okay, so like see how this person drew the circle, and then this is how big that cell was. And then they could see about 2 fit in that area. Okay, you have to draw your circle. So you're saying 7 fit, right? I think that would be fine. I would still like to see you draw the circle and draw the circle.
You just gotta search up the.
But 7 isn't a bad estimate. So if it is 7, then you would do 1.6 times 7, and that would be this. Okay? Yep, this. You're at 10x. Yeah, it's exactly that. Sorry, that's fine. Yep, so this would be your field of view at high power. Yep, just calculate that. Okay. So you are saying 1/6 fit. Okay, so you just do this. Here, use this times 1/6. Yeah. You're almost there. Ricky, you said you're done? You have all the information. Also done? Thank you. You have the stipple. Let's keep going with our notes. All right. Scale. Is that part of your notes? Yes. I have to check math later. But if it's wrong, you can answer. Okay, guys. I'm gonna say a couple more things. So go back to our quiet working. And do you have a hash to turn in? Okay. So with the scale, up top, I should have something bolded. Your scale is going to include how many times larger the drawing is compared to a life size and labeled scale bar. I know. Well, it's not the whole thing. Scale bars are very important in your drawings. So on any good IB drawing, we need to include the scale bar. We are going to have a set of questions where we will practice using a scale bar to calculate the size of an organism. Okay, but right now you're just writing the top bold. Okay, the equation, we'll go over that when we do our example. Is that our last box? Yeah, so they're all filled in now? Okay. In the scale, you're just writing the bold up there. You're good? Okay. Is this done? Does anybody else who's not on the microscope need a microscope? Okay. All right, are we done writing scale? Okay, here's a wonderful example. So looking through our boxes here, so one thing at a time. Does it look like they used a pencil? Well, we can't really tell. We're gonna say yes. Yes. Positioning. Does it look like it's in the middle of the paper? Good. Size. Is it about half the size of the paper? Good. Labels. There is one wrong thing about the label. Which label is wrong? Not about what, all of them are correctly labeling, but the format is wrong. Read your label. Horizontal. Which one's wrong? The nucleolus is not horizontal. Okay, so that one, we would make that horizontal. Okay. Otherwise, cytoplasm, nucleus, chromatin. It looks like they used a ruler. Horizontal, it looks good. Technique. Do you see feathery lines? Do we see shading? I didn't go over that. The difference between shading. I kind of did. I said you have to stipple, not shade. Not everybody knows what stipple is. If I have something that's darker and something that's lighter, shading would be like this, where you're kind of coloring in. We do not want shading, because that might show you, that might look like a structure, right? Each line that you draw, you're like, oh, that's a structure. But instead of showing a structure, what I want to do is show that it's darker. So what I do is I dot. This is stippling. And the more dots you make, the darker it'll look, right? So stippling. And if I want it to look light, I'll put less dots. So you can see example of stippling in this picture. Not all drawings must have stippling. Okay? If you're gonna shade, you have to stipple. So notice that their cytoplasm, they stippled lightly, and their nucleus, they stippled darkly. Okay. Accuracy. Well, you can't really tell that because you don't know what they were looking at. Title, let's look at the title. Notice the title is very specific. You don't just put cheek cell under 400, right? It's nice and formal. So human, Homo sapiens underlined, cheek cell, stained with methylene blue. You may or may not know that. If you don't know that, you wouldn't have to write it. And viewed under 400x magnification. That's pretty much the format you want to use. Viewed under blank x magnification. If you don't, then you wouldn't put it. If you were on a Chromebook, you should look it up. If it's an IB test, they might give that to you. So it's just about, if you have the information, you have to make sure it's in the test. If you don't have the information, you write what you do have. Good question. No. I just would like to know what it was. You can. Yeah, they're out somewhere. All right. Also, our last thing, a scale bar down here. This scale bar is showing that this line is 40 micrometers. Okay, so we'll talk about how we're going to use that. There's another actual drawing. Looking at this, which of these do you think is not correct? Go through your list, checklist, what looks good, what looks not good. Can we identify things that are not good? What? Which line? Oh yeah, vacuole. It's not, it's horizontal, but it shouldn't have that little L part. Okay, good. Yeah, I wouldn't go with a question mark. I would just either be confident or not. Yes. It doesn't necessarily have to be shaded. If you just want to go with just non-shaded, that's okay. Yes. Right. Good. So I would say this title is not complete. You should always know what magnification. If you are drawing it, you should know the magnification, right? So you always should have that in the title. Good. So title, it should include the fact that this is a single cell from the LOD leaf as viewed under a 400x microscope. Also the scale, we're going to go over scale format again. This one, good or not good? Not great. Please don't let your drawing be this messy and icky. All right. It does look like they used the right materials, though. They used a pencil. The positioning was okay, and the size was okay. Everything else was not. It's too messy. That should not be, if that maybe is your rough draft, that should not be your final draft. Here's another one. Again, the title isn't great. The rest is pretty good. Oh, the shading. Do I see shading instead of stippling though? That should be stippling, not shading. The magnification is not in the title. It should be. Well, it's like they have two titles, which is probably not good, right? Drawn at 400x life size. I would put seen under 400x magnification would be better. Okay. Last thing we'll write today is this, define micrograph. That is underneath your data table. Okay, I need happy. Are you with for that? I would rather when we actually do a drawing, I would rather because it says demonstrate. So basically, if you're drawing, make sure. And then I said pick like two of the best drawings in your book, or if you guys call them. Yeah, yeah, that's good. Yeah, I would say I would think like that first. That's it. Like we're gonna do a drawing, an official drawing. How do we need to finish this? Well, I don't know what your role looks like. And was your fill to do a triangle? Yeah. Not a circle?
Well, this was
So where was your circle in relation to this?
well, no, because this is pretty big, so the lowest magnification is pretty.
So that's the thing I need to see is like, I need to see your state and the circle around it. Because or else we can't figure out how many would fit. do you have? Alan, you only have 3 minutes. Just say.
Use mine, Alex.
The field of view should be 4. James? You? All right, we need to get those microscopes put away. We have another period coming in here. We need to be cleaned up. So let's. No, because it's English. It's probably a cross section of the root. All right. If you are on microscopes, I do hate to do this to you, but it would be great if you could take a picture of someone's notes and copy them later. Okay. Or you can use the slides and do that, but that would be a micrograph. Yes. Guys, we need to get the microscopes put away. Put your stage down. Go at low. Hold on, Alan. I'll help you in a second. Put your cards away. We gotta get cleaned up here. And you're gonna want to take a picture of someone's notes. Mark, Ian, did you guys get the notes? No. So have someone take a picture. Oh yeah. What was your question? Wonderful. What about David? Is he ready to turn it? So I would say as one of those. If it's done, put it on my desk. That would be okay. Yes. Okay. So oh, they're also yeah. Do that, but like Uriel is like starting to wake up. Also, and what's his name? Benjamin. Yeah, Benjamin. You're gonna say, oh, you're gonna just say both times, right? So I would say, like, this is like your whole circle, right? Okay, so I would say just from okay, to figure out how many of these would fit, draw your, and then you do 4 times that fraction. Okay, you do that to me later. You will be able to use a calculator on the test. Okay, because I need I forgot how to do the no, I can't. By hand? By hand. I don't remember. Oh my god. So I can turn mine in. Okay. Either later or tomorrow. Yeah. We're not doing the official title on that. Yeah, that's perfect. go for that title, that's awesome, but it's not, well, that won't be required until our next. You're drawing like you. Oh, it's cool. No, the first one's good. So did you do one? Well, how many of these fit?
I would say maybe like up to 2.
So you did 1.6 divided by 2.
Because it's just got the long.
Yeah, I would say if you were to draw another circle this size. Yeah, I would say 2. So this divided by 2 is this, right? How many 2? It's okay. More about you know what you're doing. Don't worry, as long as you know the process. And your freshman nature is beautiful.
I don't like mine.
I love your cow.
I like my cow. I don't like mine.
Well, you can always do another one, but it's good for now.
Or I'm not gonna.
Hello. Is it long or short?
No, it's actually good. So,
I've never read it.
the master of it, how it noted is,
The Mask of the Red Death.
yes.
It's very good. I'll have to read it sometime. I'm sure Mr. Tennant has read it.
Oh, yeah.
Construction paper?
Yeah,
I know, I know.
She said she was bringing, and she was bringing your Sharpie markers too.
Who?
Bronca, so maybe she forgot.
Oh, she got some of the ordering?
Yeah.
Okay. I was like, we have to- hopefully it's the neon paper.
For what?
I think I want a neon paper too, because sometimes I like to print on color
Oh,
paper.
did you put on the list?
I
put
can't
red
remember if I
construction
put construction paper or neon paper. Either way, something like that.
Yeah, clarify the calculation. So the low power is always gonna be 4,
For that microscope.
right?
Yeah.
And then to calculate the high power, how did they do that?
That's low power diameter.
Which is this one?
Yes, times low power objective. Divided by high power objective. So for our microscopes,
So
it
it's gonna be
4 times,
which is still 4, right?
yeah, the objective is 4, yeah. And then divided by
This is 40.
40. Yeah.
Or let's do 10.
Yeah. Yep, so
10.
that'd be 16 divided by 10, 1.6 millimeters.
So then thinking of the diameter would be,
The diameter of an organism.
yeah.
So if we had an organism that took up, let's say 3 of them would fit, it's 1/3, right? So you
Of the
just
whole
take that of the whole thing. So we know the whole thing is 1.6. But we only want 1/3 of the
I only
1.6.
want how many
Right.
the diameter.
So it'd be
gotcha.
1.6 times 1/3 or divided by 3. Yeah.
Oh, okay, that's easy.
Yeah. It's like, this is definitely one of those things where I feel like they just have to do it enough. And then it's like, oh, that makes sense. And in the notes that she actually explains it 2 different ways. There's like 2 different methods. So first method, that's the fraction one. Because I feel like that conceptually makes sense. For the fraction. The other one is just divide the field diameter by the estimated power. It's the same thing. That's why I kind of just taught method 1 this year. And then in the notes, she has like an example of this. But I just am going to use that as an example. I think I might change that. I think conceptually the fraction makes more sense in my mind. Because to do this, basically you're doing this.
They're the same.